Hello! Oh my goodness. How are you?
How are you?
Well,
I'm balanced on an emotional precipice right now.
are you?
That song,
Oh,
all of their songs make me very happy.
and?
Sad.
Yes. Wonderful. Thank you for sharing, and I feel you with the gifts. If you are feeling Kermit because of the topic test,
It's a Kermit Lane distribution.
yeah, there are many things that you can do to reduce that feeling. Now, that feeling isn't necessarily a bad feeling. Being anxious or nervous, if you can learn to harness that and use it as a means of focusing, it can actually be really powerful and really helpful. And so if you're feeling Kermit about the topic test, there are a few things that we've got for you. So just a reminder that we do have that asynchronous study tool that's open, and you all are a pretty darn good group because it is looking really good.
Gosh
Darn it. It's looking really good. And the more people that are on it participating, the better the tool becomes, right? So this isn't an opportunity for us to write good questions and things like that, because that's not learning. I've done the research on it. It doesn't help. But this does, and it takes a little bit more work, a little more concentration, and all of that is, consistent with studying and learning and all of those things. But already, many of you have been on it and have been contributing to it, and it looks great. And we will continue to monitor it and jump in, and do all of that stuff. So
it's not just an Elaine and Kermit. It's a Nathan Apodaca as well.
Where? Yeah. Is that just chill, just coasting? Is that what that means? Wonderful. Yay. Okay. Now, Elaine down there in the bottom corner, suggests that you might be overwhelmed, with all of the stuff that you've got on your plate, and not just school-related stuff. And there are ways of managing those things. And tools that you can use. And man, I'd love to, maybe we could do it in office hours and student hours afterwards, maybe talk about some of those things. And again, there are these tools where you don't have to shut down. Are you getting some comments, some helpful comments?
You can't spell studying without dying.
Oh no. Oh no.
I mean,
no.
Yes.
But no. But no. Okay.
We spell it, there's a silent P, and I have some Welsh heritage, so there's two L's. And they just move around.
Yeah.
Do you see the solar system that's
I do. It's stunning.
Yeah.
Let's screenshot that, because
Mercury, Venus, or Mars, Jupiter.
Beautiful. So overwhelmed, you're saying. Yes, I hear you. And there are tools, and one of those tools includes letting stuff go as well. Just being comfortable with not doing all the things.
I'm trying so hard not to sing.
Don't.
Let it go.
No. We're kind of into Frozen right now with our kids. Let's not do that. No. Okay. Your solar system is stunning.
Yeah, one more
thing?
Yeah, one more request is to say out loud where the asynchronous study tool is.
Yay! So all of the information on Course Link. Check it out. There is an announcement with a link that you can go there and get studying. And honestly, the less time you spend reading over your notes, not to say that that's a bad thing, the less time you spend doing that and the more time you spend on this asynchronous study tool, the happier you will be. Of course, it's not going to cover 100% of the things, but the main things are there on the asynchronous study tool. And what you want to do, so if you haven't been there yet, you don't know this yet, but the idea is to write statements, whether they're true or false, it doesn't matter. What you want to do is you want to write a statement that half of the students, the other people, think is true and half of them think it's false, right? You don't want to write something so obvious that it's easy. So you want to write tricky statements, not tricky as in oh, I had a spelling mistake in here, so it's false. No, challenging statements. Yes, that's what you want to do. Okay.
You can't spell statements without living. If you spell it wrong.
No. Okay. the solar system is
Maybe one final question from the chat is that we talk about the seminars for this week and next. What's up? What's going on?
check your schedule and check Dr. Ward Campbell's announcements, because if I tell you, I will probably get it wrong.
Yeah.
So please make sure that you check in with those. The regular announcements are posted as well as the schedule, and it's all set up there. So check it out. I don't want to get it wrong. All of that stuff, the seminars and the quizzes and the course link and the stuff, that's all Dr. Ward Campbell's area. We are 3 instructors for this course for obvious reasons, because there's a lot going on. And I don't want to get it wrong. Okay.
Should we start?
Let's start. I think most people are here who are planning on being here. Okay, wonderful. I don't want to clear.
Got him.
Okay.
So when we say start, one more review thing.
One more review thing?
When you go...
Right.
And that is just to remind you that there is a playlist of all of your... Certainly you get all of the lectures. They're all there per lecture, per week on CourseLink. And if you follow that up, you'll see a playlist that just keeps growing and growing of all of our classes together.
There you go. Thank you for doing that. That's great. Okay, we're going to do some stuff. It's listed here. You can take a look at it later, because...
Yeah, because look at that first one. That's a big deal.
That's a big deal.
That's all the time.
It's complicated. Okay. Why study bio? We're going to do this snappy, okay? Because we have like a lot of time, a lot of things that we have to do today. Yeah, basically, I'm just telling Smith to not get too excited to like.
Because I want to talk about this. This is But why study biodiversity? What is biodiversity? What it is? What it isn't? We talked about that a little bit already last week, and you guys had some great ideas. But it is, of course, a growing area. An umbrella study in biology that we said earlier, you can do it with gene sequencers, you can do it with field guidebooks, and you can do it with satellites. It's a contraction of the phrase biological diversity that is not that old. E.O. Wilson did it in the 80s. One of the common metrics that we use, of course, is to count the number of, what is biodiversity? Well, one measure could be, let's count the number of species.
It's a good start, right?
It's a good start. It doesn't have to be the end. But then, as biologists know, that it's a good start, and then you're like, oh. That's right. It's like, this is harder than chemistry. I had a great friend when I was doing my PhD that said, oh, well, don't worry about this. It's biology. It's not rocket science. And I grabbed them by the shoulder and shook them, and I was like, I know. It's harder than rocket science. Rocket science is easy. It's simple Newtonian physics. We don't have 36 different concepts of what a meter is.
And we have 36 different concepts of what a species is.
Which is okay.
Which is okay, and it's fine, and whatever camp you fall into, and you probably are good with like 5 or 6 different definitions,
because
right?
It depends.
It totally depends, right?
This is a common one. It's probably the one that you've been introduced to before. It's probably the one that if you talk about this kind of thing at Thanksgiving, species that are actually or potentially interbreeding, that are reproductively isolated from other such groups and producing fertile offspring. So we've got the picture there of dogs. Impossibly different morphologically because of artificial selection for hundreds and hundreds and hundreds of years, but yet still members of the same species. That if we had a large ladder, they might
be able to
not
interbreed.
Be reproductively isolated anymore.
This is definite reproduction isolation for sure.
There's a whole bunch of whatabouts, and a great one popped up in the chat, which is what about polar bears and brown bears?
Yay.
Hybrids are a big deal, and that specific one, file it in your brain. Let's pin it.
Let's pin it.
Pin it on the wall, because
we're going to
spend
a lot of time on that.
Spend a bunch of time about that.
Yeah.
But what about asexual species? There are species of mite and rotifer that have not had sex. It's like a long dry spell. Like we're talking like centuries. Like centuries. And what about hybrids? The brown bear and the polar bear. Won't someone please talk about the Fowler's toad and the American toad? I will. I won't, because we're being peppy. What about connected extremes? What about species that are described at one part of the northern hemisphere and their range extends all the way around the pole? But as you travel around that range, you see differences. And could the one in Siberia actually interbreed with the one in Churchill?
So if you're curious about what we're talking about, check out gulls.
Yeah, and then leave.
And then
Gulls,
don't
who
call
cares?
them seagulls. You call
Please
them
don't.
gulls.
I get so irritated when we watch closed captioning and they're like, seagulls calling. I'm like, impossible!
They're just gulls. There's no such thing as a seagull.
No.
Okay, and here's a paper that got the number wrong and said that it was about 26.
It's old. That's all.
Ah.
It's just we come up with some more. 8 years? 9 years?
9 years! So, please, if you're thinking, do I have to know the number of species definitions? No, because no one knows the number, right?
Well, take a grad course and then sure.
Yeah, what you need to know is that there are a lot and that they focus on different scales of organization and that they have different implications and that they don't apply to all of the things all of the time, right? That's the complexity. That's what makes biology what it is. This sort of ability to be comfortable without knowing all the things.
And in 1 second of a philosophy class, species aren't Aristotelian. They're not like hard-edged, like it is or it isn't, and that's because of time, because speciation takes time, so they're fuzzy. And species are literally fuzzy, even the shiny ones, because you can be a bit of one and a bit of another.
You totally
and that's
can,
okay.
and that's okay.
Because that's how life works.
Uh-huh. And all of these ideas need to be applied also to the human populations and the human species, right? Good. Okay. So forests, back to forests and biodiversity and all of those things. Get a browser open and we're going to go to Menti in just a minute. But before we do that, just a reminder of what we're dealing with. So the idea is that we have these 4 woodlots, these 4 places that are different. And in trying to figure out and explain, first of all, what that difference is, and then more interestingly, potentially, isn't more interesting, why that difference is, right? These 2 things are really important, and they do kind of characterize ecology and the tools that we use in order to be able to do that. So we'll show you some tools today, some good tools that are helpful and not all encompassing, right? So hopefully you'll be able to see where the pros and the cons are of each of these tools. But let's go to Menti, if you don't mind. We ended off asking you this question: what information do you still need? So for like the first time ever, North Campus Ravine didn't do as well as it usually does in the first round, but Browns Woods also getting a beating. You were kind of between the 2 in terms of which one you would get rid of if you had to. And then, of course, Camp None was growing as well, which is totally fine. What do you need to know? What more information do you need in order to make that decision?
I see you in there and some great things already.
Wonderful. This person is like, we see you. We'll share that in a second. Just keep on going. Keep on sharing.
If that keeps up, I could do a lecture on an actual lecture on papillons in New
Look. Again and again. Yeah. Okay. Good. Thank you. Wonderful. Well done. We'll wait just a few more seconds to get your answers in. And here we go. So, just taking a look at what we've got here. Somebody very passionate about moths. Okay, so who lives there? Biodiversity. What species exist in that habitat? The level of biodiversity. Species rarity. So are there any rare species? That's delightful. What else? Lots of species things and basic. I wonder why they're saying species. We've been talking a lot about species, and this is great. What habitats are there? So I like that one a lot because it's like what else is available.
I like seeing people pop up ideas about rarity or frequency.
Yep. Species type, invasive or endemic. I love that.
I saw endangered somewhere.
Yeah. That's really good. Elevation
There's a
differences.
history as well, and some element of cultural. There was like who's living there.
Yeah, this one I love, and you're laughing, but oh my goodness, are you ever right? You know, like is whatever it is that we're gonna do with that land, is it appropriate for the planning? Now, clearly, condos and other activities may have different levels of importance to us and value to us. But no, it's a real thing. It's a real consideration. And I think too, one thing that in that respect, a history of land use is important. Either whether it's a biological history. So you've touched on the biological history of those places, right? With respect to the number of species, and it's also important to take a look at the human history of those places and what the relationship is of those places to the people who lived there or used to live there. And so, yes, we need to sort of take into account all of the things. It is a large system of consideration. So this is systems thinking, and we need to understand, that piece of land from all of these different system perspectives. Super.
History doesn't have to be ancient history. There's a big catastrophic perturbance that has happened in eastern North America to forests with the arrival of emerald ash borer, and we lose lots of ash. So who has more ash?
Yeah.
Where are they? Because when we started doing this unit, there were a lot of ash trees, mature ash trees in all of these woods, and there aren't anymore.
Yeah.
They're literally all gone. They have died and they have fallen down.
Yep. For sure. Sorry, that yellow slide just means that we were going to be working on it and we forgot to hide it. So here are some things that you said. Not all of the things, but most of the things, which is really good. So I added the historical significance and all of those things. So here are some more things. And what we're going to do for today is focus on the one in red, number of species, whether they're endemic, invasive, endangered, all of those things. But right now, we have to figure out how many species there are in each of these woodlots, and that is a challenge.
It's a challenge. So there's lots of ways to measure diversity. Yes, using species as the unit that you're going to count. Okay, but you're not done yet, because then you have a simple question about So is it just the number of the species? Does the abundance of each one of those species matter?
Yeah.
How are you going to compare between places? How are you going to compare in a region? And we have names for all those different kinds of diversities because this has come up before.
That's right. Names and math all together. Oh, so good.
We got writing and arithmetic.
So
right up, eh?
yeah, so these are sort of classic ecological concepts, right? And we have these like ways of quantifying or describing the biodiversity of different places. And we can do that within one habitat. And again, what is a habitat? You need to define it. And when we do that, sometimes we call that species richness. And there are many ways of quantifying species richness, and one of the ways that we do that is using a concept called alpha diversity. We also can look at species evenness within a habitat, and one of the many ways of doing that is by using the Shannon Diversity Index. We're going to introduce 4 indices to you. There are many. These are the ones that are most helpful to understand these larger concepts.
There's literally a book.
Yeah. Okay. Now, across or between habitats, because sometimes it's really important to compare. And sometimes it's really important to compare not across geographical space, but across time, right? So we want to look at either 2 things side by side, or we want to look at one thing over time, okay? And to do that, we can look at species richness difference. And we do that using beta species diversity.
Jacquard!
Or we can look at total species richness. And this one is super fun and super easy, but like also interesting. And we do that using gamma species diversity, okay? We will go through all of these, but here are the questions. Here are the big ones, right, that we want you to focus on as you learn these things. What are these and how are they calculated? I am not interested in you memorizing a formula. No. We want you to understand the variables that matter and what happens when you tweak a variable and what that means biologically, okay? Which variables contribute to changing these calculations? How can I change from a biologically important perspective the Shannon Diversity Index? Right? What happens if this happens, and what, will the number go higher or lower, and does that matter? Are they correlated with each other, or can they vary independently?
We really have to pump up this ball.
I'm sinking. I'm sinking on my yoga ball. Okay. So let's, again, we're recording this. This is like going to happen pretty fast paced, but just to give you an example of how we use these numbers. Okay. So imagine we have two very simple habitats. Habitat 1 and but you could just look. You don't even have to imagine. Ooh. Look at the screen. We have two habitats, Habitat 1 and Habitat 2, and this is what they look like with respect to their species diversity and abundance. Okay? So, alpha species diversity is simply the total number of species within a habitat. So tell me, please, represented here in Habitat 1, what is our alpha species diversity for Habitat 1? There we go.
Unos, dos.
There.
Tres.
Thank you, Liv.
Yep. And
great.
You got some good answers in the key.
Okay.
In the chat.
Good. Nice. Thank you, Jacob. I see you. Wonderful. Yes, easy. That is alpha. That is it. It's complicated, but that is it. Habitat 2, what is our alpha species diversity? Thank you, Catherine. Thank you, Jacob. Thank you, Mitchell. Yeah. Good. Great. Okay. Alpha diversity. Again, kind of easy, but also complicated, and we'll tell you why it's complicated. But the alpha species diversity for each of these habitats is 3. Now, are they the same?
I think you can even see just looking at that going, okay, so this is a metric that in these two places gives us the same number.
Yep.
Maybe we need a metric that reflects some other things.
Some other things. But
those don't look the same.
Thinking a little bit about even just calculating the number of species in a habitat is complicated. I'm like, oh yeah, super easy, and then buzzkill, complicated. But here we go. Because we may very well be able to count the number of species within a small 1 meter by 1 meter little square. And even then it would be difficult. But if we focus on the plants, or we focus on, the mushrooms, or we might be able to do it in a way that's satisfactory. And we know that the more times we try, so the number of plots that we sample, the more species we will find until a certain point, right? And so what you do is you start out with okay, I'm going to go and look in 5 different places. I'm going to count the number of species, and then I'm going to increase it to 15, and I'm going to find a few more species than what I found over here. And I'm just going to keep on going until I reach this plateau, right? Okay, that makes sense. Totally fine for the Arboretum. The problem is that when we're talking about a large region, like, say, Southern Ontario or Canada or North America or the planet, we don't ever really seem to get to that nice plateau where we can be way more confident that we have found all of the species that are in that area. And so scale, geographic scale, really matters when we're talking about exactly. There's so many, right, Owen? So yes, it gets really complicated really quickly.
And that gets really complicated really quickly at scales. So as you deal with big things,
yeah,
this is a pretty easy, it's a good job. It's like you could go out and do the Arboretum plants, but if you were to do the Arboretum colembla or the Arboretum cetomygid flies or the Arboretum mites, it would be much harder, and it would increase and take a lot longer to reach that asymptote.
The Arboretum mammals?
You should be able to do.
We could do.
Yeah.
Okay, cool. So, how do we know when to stop counting? The answer is when sampling intensity is saturated. So these are some words for you. Basically what that means is you're reaching this plateau. You're like, yeah, I've probably got the vast majority of the species counted once you start to see this plateau.
Yeah, I just used the word asymptote
Asymptote.
as a synonym to that plateau
Yeah.
idea.
Okay. So it's in yellow. I'm going to throw this at you. It shouldn't be in yellow, but here we go. Launch. We're going to throw a question at you. Here you go. Take a look. Sorry, I forgot to make it not yellow.
That's right. Yellow is bright and
bright.
looks well on film.
Yeah.
It brings out the jaundice in my eyes. I am struggling because you have said complicated so many times.
I'm sorry.
And I want to sing.
Why do you have to go and make things so complicated?
See the way you're acting like somebody else gets me frustrated. Life's like this one. You fall, you fall, you fall. So our oldest son was when we sing that with him, he drops to the ground. He falls, then he crawls.
Okay, sampling intensity is almost saturated at OG. Sampling intensity is saturated at AY. No. KR is likely to be oversampled. Richness is lowest in SM. Diversity is highest in AY. Okay. Super. Okay, just a few more seconds. You're doing really well. Good, 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 good. Go, 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 go.
I always wondered what was he yelling for. Just lay back. It's all been done before. Dr. Smith, do you like ants? This seems like a leading question.
Oh no,
Let's change
no,
everything we're gonna
the
talk about.
can of ants open, can of worms. Okay, so we're going to close it down in about 10 seconds. Go ahead.
Like a lot.
Like a lot. Okay.
We talk about Avril Lavigne in 2700 if you didn't get enough.
Oh,
Because
no.
she got Lyme disease.
That's right. That's
As
right.
did the Beebs.
Ah,
We have a Canadian musicians infected with Lyme disease lecture.
okay. Share results. Here you go. So, split between A and D. The correct answer is D. Yeah, so sampling intensity is almost saturated at OG. That's great. You can see that it's really starting to plateau there. You might even want to say, you know, saturated, but it's like definitely almost saturated for sure, okay? Now, diversity is highest at AY? No, not at all. Right?
Now why? Because
Why?
Why
You want to
in
pop it into
AY?
the chat? Why?
Yeah, why is, tell us about why.
And tell us which other line makes that a false statement.
Yeah.
Which other line? You can draw on the slide
You
too
could draw
if
it
you
as
want.
well.
Yeah.
That would be great.
What is sampling intensity? That has to do with how many plots you've sampled.
Yeah, here we go. It's like something out of Monopoly. It's the old KR line.
It's the KR line.
It's
Very
the old
good.
KR
There,
line.
Danielle. Super good.
Nice.
So
well done.
So yeah, Alyssa, take it forward. Keep on going. What could it be?
You guys got this. And what does saturated mean? We're talking about
Yay!
asymptote or plateau.
That's right. That's right. So because not all of these lines have plateaued, we don't know what the diversity is. We know how many species have been found within that meter squared, but to then use that as a proxy for, thanks Danielle, that's great. As a conclusion without it being saturated would be premature.
So is it highest in AY? We don't. It may be.
It may
We
be.
do not know yet
do
because
not
of the
know.
old KR line.
Because of the old KR. Exactly.
Woo-hoo.
Okay. Cool. Thank you. That's great.
Now we'll wipe off the board.
Yeah,
That's
good.
not how that works,
That's
is
not
it?
how that works. No.
I mean,
Okay. So, is alpha diversity the number of species The only important factor is species richness, the only important factor. We found in our original habitat that it was 3 over here and 3 over... Are we done? Are we good? Should we just like, that's it? We can make decisions? Or is there something else going on? Well, clearly these are not the same. Even though they have the same number of species. So what we want to do is try to figure out how else we can sort of quantify or describe what's going on. And we do that using a concept called evenness. So the more even something is, a habitat is, the more similar number of individuals of one species there are in the habitat. So, you know, there are 50 individuals of species 1, 50 individuals of species 2, 50 individuals of species 3. That would be remarkably even. And weird, right? But we have that going on in habitat number 1 on the left-hand side, right? We have 2 individuals of each.
We have 2 deciduous trees, 2 conifer trees, and 2 sideshow bob trees.
Yes. So high evenness versus something that is low evenness, where we have one species that's kind of dominating, and then one species that's only present as one individual. So the range of the number of individuals of each species is broad, which means that it's lower evenness.
People want to know, who is Shannon?
Ha!
Was she an Irish diversity biologist?
No, and as much as I'd love to say
that
she is a she is not
Shannon
a she.
O'Carroll.
Yeah. So how do we measure this? We can do this using things like the Shannon Diversity Index, which basically was made by the Darth Vader of ecology. So Claude Shannon, old white guy extraordinaire, came up with this.
This is his birthday. He'd just blown out candles.
Not a happy person. And came up with this particular equation. Now, this equation, literally, my face, is the face ready?
Now, just wait. The face is ready, but you got to get through because there's a Star Wars correction factor.
Did I make a mistake?
Well, no.
It's Darth Vader.
It is not Darth Vader. It's actually Grand Moff Tarkin.
Oh, no.
Like, is that not the same guy?
Yeah, it's
Okay.
the same guy.
Now, your face, when doing the Shannon Diversity...
My face looks like this. I promise you, when I see math equations like this, I'm like, no. But then, like, all you do is you print out that math equation and you put it in a place where, like, your brain is, but not focused. So, like, on the fridge, like when you're going to get food, or in the bathroom, right? When you're, and then eventually over time, your brain works on it without you knowing and without it hurting.
Try and make it a sentence.
And then you make it a sentence. And this, to me, is a lot more clear, believe it or not. Okay, so don't panic. Basically, right? One of the things that you can see in an equation like this is this huge thing, which basically just means you're adding stuff, right? The sum of, the sum of what? Really simple stuff, right? Shannon Diversity Index is basically what you're doing is you're taking the number of individuals of species 1, you're dividing it by the total number of individuals of all of the species in that habitat. You're multiplying that by the ln. Don't worry, it's that whole log, ln, blah, blah, blah thing that you've got on your calculators. Like, who cares why we do that? The same number as this one. So it's basically just adding up the proportions of individuals of one species over all of the individuals of another. And then you move on to species 2. And you do exactly the same thing. The total number of individuals of species 2 divided by all of the individuals, just like that number over there. And you multiply that by the ln of the same number. Again, who cares? Why? Because Darth Vader decided, or what? Who? Grand
Moff Tarkin.
Okay. Grand Moff Tarkin decided it would be fun to do that, but who cares why? And we just keep on doing this the whole time. So it's basically just working with the proportions, right? And that's what evenness is the proportions. Don't panic. Practice it though, and like bring your brain to that uncomfortable place.
I think there's a group of people on the other side of this Zoom camera that are ready for this, because there's been a lot of questions in the chat about percentages, and when we say proportions, think percentages. That's fine. And this is Molly Shannon, Claude Shannon, thinking about how to incorporate that.
Yes, and this particular equation was not used for ecology originally. Claude Shannon, did you know this? Mm-hmm. Claude Shannon came up with it as a way of breaking code. Because what you can do is you can take a look at the evenness or the proportion of different sort of symbols within a code to start to break it down. Did I explain that right? Yeah. Okay. And then somebody was like, oh, we could totally like add this to ecology. And you're asking about the minus. The minus is simply to make it a positive number.
And so that we could be more intuitively going, okay, if it's more, instead of having to think about it on the bottom side of the graph, let's think about that on the top.
There's no reason except for our brains like to have positive numbers more than minus numbers. And if you add up all of those things with the lawn transformation, you end up with a negative number. So if you make it a negative, you get a positive, and that makes us feel better about ourselves.
It's feng shui. It's like, how do we want to look at this? We want to move this chair on the top part of the graph.
That's right. Okay. So, just to review, within habitat. So we're only dealing within habitat for the moment. Species richness, number of species, alpha diversity, for example. Species evenness, Shannon diversity index, for example, where we talk about the relative proportion of species within that habitat.
Some of you thought in there that you'd seen equations like this before when figuring out standard deviation, and you're not.
All good. Yep.
You're in the right space.
Very good. Okay. Now, what about when we need to compare two habitats? Because Shannon diversity index is great within one habitat, but as soon as you start adding more and more species or all sorts of things happen, it doesn't help very much to compare across. So how do we take a look at describing species richness across habitats? One of the ways that you can do it is by going, well, actually, it's all just one habitat. And what we're going to do is we're going to count the total number of species across both habitats and do basically an alpha, but across multiple habitats. And when we do that, we call it gamma. So we have total number of species across all habitats. We've changed it up here, so it's not the same 3 as before for alpha diversity. We've just kind of changed them a little bit. It's still alpha diversity of 3 in habitat 2 and habitat 1. But now, because we've made it a little bit, these are different species, when we do gamma across both, you don't just add them because they share a common 1, right? So you add them as though they were one habitat to get gamma diversity of 5. So another way of saying this is that we've basically just increased the geographic scale of our alpha diversity calculation and made it one habitat. And it matters. You'll see. We have homework for this. It's helpful. You'll see why it matters. Okay. Beta diversity is the last one. This is it.
This is the best one.
You like this one.
I rank them.
I find it the most useful. Is that possible? Especially in the context of the climate crisis and all of the research that we do. Beta diversity.
Space and time.
Yeah.
Our questions are always going to be about space and time, about how is it changed in space or changing in space or through time.
Yep. Good. You want to do?
Sure. So we've landed here again in our Magic School Bus outside of Species Habitat 1 and 2, and we see that there's a sideshow bob tree, some conifers and deciduous trees in Habitat 1, and then some cheerleader pom-pom trees and some other coniferous trees in Habitat 2. And we can calculate the alpha species diversity for each. We can calculate then the number of species that are in common. And this is the common. So again, just like we talked about with the why was there the negative, you'll see if you dig around in this.
Yep.
Beta diversity can be reported as a negative or a positive. It really doesn't matter because what it's measuring is kind of the strength of overlap in between your two places.
Yep.
So you could move these kind of two habitats roughly circular and think of them like a Venn diagram.
Ooh.
And then your beta diversity is in the middle there.
I like that.
Yeah.
Okay.
Well, and it works even when your habitats aren't circles.
It kind of does what gamma doesn't. It's kind of the opposite, right? Where gamma sort of highlights the things that they have in common.
All the couldas.
That's right. And beta highlights the things that they don't have in common, right? Yay. Okay. So it's a measure of how different the diversity is, right? Remember, we're still dealing only with species. We're not dealing with evenness. We're just dealing with numbers of species. So what you want to do is set up a really simple equation that doesn't look, why do we have to make them not look simple? Like, can we, I don't know. But
I hang on a second. I think they're pretty.
It's pretty simple? Okay.
Yeah.
So what you want to do is beta is equal, to breathe. Beta is equal to the alpha diversity of habitat 1 minus the number of species that they have in common between 1 and 2. Okay? So you're basically removing all of the ones that they have in common. Plus the alpha diversity of habitat 2 minus that same number of the number of species that they have in common. That's it.
Yeah.
Okay? So kind of nice and simple, right? Especially if you put it into words that make sense to you. You don't have to worry about memorizing this. Oh, so what is beta diversity of these two habitats? Tell us.
There's some answers popping up on the screen and in the chat.
Wonderful. That's great. Thank you. Yes. Well done.
Some are declarative, some have question marks.
Yes, that's fine.
4.
4. 4? 4. Yeah, 4. Okay. Good.
And
Wonderful.
then a question of, are red foxes native to North America?
Yeah.
4.
Foxes sleep in the forest.
Yes, they do. They are.
Lions sleep in their dens. Okay, wonderful. Okay, so to review, then, when we're talking about it across habitats, right? We're talking potentially about species richness, and when we do it across different habitats, we call that gamma, but essentially it's alpha. But remember, you're not adding the alpha of one to the alpha of the other, because you'll be double counting if they have any species in common. Okay. Number 2, species richness difference. And that we use beta to do it. And remember, we can do it over time, so we can calculate beta from the same habitat 10, 15, 20, 30 years apart, or we can look at it between 2 different habitats.
Yeah. So gamma and beta, think about the species in common, because that's around which it fluctuates. You want to accommodate for the species in common, so not double count them with gamma, and you want to zoom in on that in beta.
Cool.
You're a national treasure. I would say international.
Thank you. Okay, moving on. So here's the homework, because I think if you're overwhelmed and you're like, I don't know what these mean. They were supposed to teach me. I was here for an hour. I paid attention, and I still don't know what these things are. You're not supposed to be able to learn all of these things in one go, and any profs who think so aren't teaching you. So this is the teaching part. This is the learning part. This is the homework that we want you to do, okay? It's not easy, and it's going to tell you a lot about these things in your own words so that they make sense, okay? So we've given you those sort of icon represented woodlots for the Dairy Bush, Browns Woods, and North Campus Ravine. Those 3. They're in the previous week's or the previous class's slides.
And they'll be in the PDF for today.
And they'll be in the PDF for today as well, okay? What we want you to do is to calculate these indices for those woodlots. One way that I like to do it this way. I like to make a table, and I like to put the 3 different woodlots down in one column, and then across the different indices for each of them. Because the ones for like within the habitat, you'll get a different number for each. That's cool. But then when you're comparing the habitats and make all of the different comparisons possible, you can use it to kind of, show, you can create like another table with the habitat along here and the habitat along here, and then kind of show which comparison you're doing. That's one way, but it doesn't matter how you do it and how you organize it. But that's what you should do.
So I have a table even before that.
What would you
if
do?
You want. It's the same, starting the same way. So your table was the habitats with the woodlots across the top.
Yep.
And then the indices values. And one of the ways to help you get there might be to put the habitats across the top and then your species and count them.
Also.
And their counts.
Oh, yeah. Yeah.
To fill that in.
So like 3 tables you could make out of all of this.
And then if you start,
Huh. Okay.
Might help you see the pattern,
You
see the forest, the
pattern.
trees.
That's right. And don't get hung up on whether or not Brown's Woods has 6 species or 7.
No.
Doesn't matter. Just commit. Just pick one and then deal with it, right? Because what we want you to do is to see the patterns. What happens when I do this? How does this number change if I do that? Okay. Anyway, if you go through this, then double the number of individuals. And some of you, you write and you're like, what do you mean double the number? I can't. That wouldn't be what this habitat is. We want you to see the pattern. So imagine that you're doubling the number of individuals in each woodlot. Do the calculations again and see how the numbers move, right? Just try it. And then when you get stuck, let us know. Go on the discussion board, work together, however it is that you need to. But play with the variables, move them around, increase them, decrease them, and see what happens. Okay, so here is more specific instructions if you want. This is a great calculator for the Shannon Diversity Index, for example. And if you go there, I believe you will see that there are actually 3 Shannon Diversity Indices, one using log, the other using lawn, something else I don't even know. Pick the lawn one because that's the one that we're using. Play around with these numbers. Don't get so nervous that you're like, I don't know what to put in this box. If you don't know what to put in the box, put something and see what happens.
You can't break this.
Can't break it. The only thing you can break is your confidence. So just play with these things. All sorts of things for you to notice, instructions, and a guide for you to go through it. And here they are for you to use, okay? Just figure out, if you call this one the same as this one, fine. It's not, but it doesn't matter. Just commit. Just figure out what you're going to, use for your total number of species. And that's
it.
Is a class that is done on time.
We've heard they exist.
No, we're pretty good. We're only usually 2 minutes over.
We're almost out. We're pretty consistent.
We try. Okay, good.
We aim at it.
Yeah. So stick around if you have any questions for us or you want to talk about something, life, the universe. One thing I did want to tell you. This is good. Okay, so yes, the university still is using Respondus, this online proctoring lockdown stuff. And I'm a senator, so I'm on Senate, and decided to just flat out ask about it. They recognize that it is racist software. They also now just recently came out, not the university, but there's been a study that was done to demonstrate that it violates AODA. So that's for accessibility and for students with disabilities. It violates all sorts of those sort of policies and requirements. Therefore, and I checked as a senator at Senate with the provost just a few days ago, we did used to have a policy where if students were concerned about their human rights and violations with respect to Respondus, they could write to their professor in advance, give them a little bit of notice, and tell them that you had concerns with respect to your human rights and that the policy of the university was such that a different evaluation or accommodation would need to be made. Now, I don't know what that difference will be and how it will be set up, but if you are in any way concerned or upset by the use of Respondus, then you have it within your right to do that. What I'll do is I will draft a sample email that you can send if you want, modify it however you need to. You do not have to declare why it violates your human rights. You don't have to share any private information. You just simply need to state that it does. Yeah, so I will prepare like a sample. I'll post it on a CourseLink announcement by the end of today if it can be helpful as we go into midterm season. We don't use it. We don't want to use it, so we ask that all of you engage in ethical behavior with respect to taking tests, and as long as that happens, we are more than happy to not violate your human rights. So yeah, work with us, we'll work with you, and we will also help you if there are any concerns that you have about Respondus in any other courses. And now we are officially 1 minute late, which means it's time to end class.
So now we must be done.
Take care, everyone.
See you in a few minutes if you're sticking around.